Hello everyone, my name is Qi Gao and I'm a postdoc in Dr. Kinfei O's lab. Today I'm going to introduce one of our recent work, indexing gene isoform quantification error by k-value. RNA-seq have been widely used for quantitative analysis of gene isoform expression. The rates obtained by sequencing could be assigned to annotated gene isoforms to calculate the expression level. However, due to the similarity between isoforms in the same gene, many of the rates may not be uniquely assigned to one isoform, which may cause error in quantification. For example, the orange rays can be uniquely assigned to isoform 1 and blue rays to isoform 2. However, the two gray rays can be assigned to either isoform 1 or 2. Those gray rays are called ambiguous rays. As shown in these two figures, different genes have different isoform structures and different level of uncertainty in assigning rates. Some genes have much higher proportion of ambiguous rates than others. Then how can we figure out which gene will have larger error in quantification? In this study, we propose k-value, which is a proxy for the gene isoform quantification error. To evaluate the estimation uncertainty and quantification error, we first construct a model for isoform quantification. Considering the difference between ambiguous rates and unambiguous rates, we learned that different rates may contain different information about the quantification. We need to categorize the rates into different types, and each type of rate should contain similar information. So in this linear model, D is the proportion of rates in each rate type. A is a matrix of probability that a type of race is originated from the corresponding isoform, and theta is the relative abundance of isoforms. After we define the rate types, B would be observable, and theta is our target. So how can we find this A matrix? Here we introduce the rate isoform alignment matrix we used in this study. For this example gene with two isoforms, we first split the gene structure by the starting points, junction points, and ending points of all its isoforms, and select that the exonic fragments. In this gene, a total of 5 fragments are selected. They are F1 to F5. Then we would use a pseudo-array of fixed length to scan over each of the isoforms. By doing this, we can figure out all the possible positions for rays originated from this isoform. The length of rate in this total example is 5. Each of the 5 connected blocks represents a rate position. Those rate positions can be aligned to different combinations of exonic fragments. For example, these rate positions are aligned to both F1 and F2, while this rate position to both F2 and F3. Then we can define regions and effective lengths. Regions are the combination of exonic fragments with possible rates aligned to, and the effective length of a region is the number of possible rate positions that are aligned to this region. In this way, we can build up this matrix of effective lengths. For example, for isoform 2, there are 2 rate positions where the whole rate belongs to F1, while there is no such rate positions for isoform 1. And there's a special rate position that belongs to a combination of 3 fragments, F2, F3, and F4 in isoform 1, and there is no such rate positions for isoform 2. Here we also calculate total effective length. And at the end, we normalize the sum of the effective lengths up to 1 for each isoform, that is, for each column. This matrix is built by aligning pseudo-rays to isoform. So we call it the rate isoform alignment matrix. And we would use this matrix in the linear model. Now we can clarify the definitions in this linear model. Now, B is the proportion of rays in each region, and A is a matrix of probability that a region of rays is originated from the corresponding isoform. So the problem becomes how to evaluate the quantification error based on this model. To achieve this, we design our k-value measure based on the characteristics of the A matrix. Let's take a look at some features of the A matrix. We simulated 100 theta values whose positions form a circle with radius 1 and center 0. Then with two different A matrices, A1 and A2, we can calculate the corresponding B1 and B2. The values of B1 and B2 forms ovals or ellipses. The ellipses have their longer radius and shorter radius. We refer to the length of the longer one as sigma 1 and the shorter one as sigma 2. In math, those radiuses are defined as the singular values of the corresponding A matrix. As shown here, different A matrices have different sigma 1 to sigma 2 ratios. Why is this ratio important? So let's continue to work on the example. Now we add the same perturbation theta B to all the B1 and B2 values. By doing this, we want to figure out under what condition the estimation of theta is more sensitive to this perturbation. And the result is presented here. It's clearly shown that delta theta, which represents the quantification error, is larger for the A matrix with larger sigma 1 to sigma 2 ratio, although we gave them the same perturbation theta B. In math, the sigma 1 to sigma 2 ratio is called the condition number for the corresponding A matrix. The A matrix with larger condition number is more sensitive to perturbation and tends to have larger quantification error. And we can use this idea in our linear model. That is, we can use the condition number of the A matrix to measure the impact of data perturbation theta B on quantification error theta theta. For our linear model, the condition number is a ratio of the maximum singular value of A matrix over the minimum singular value of the A matrix. However, some matrices may have singular values equal to 0. To avoid division of 0, we define our measure, the k-value, as the generalized condition number of A matrix. Compared to regular condition numbers, we replace the minimum singular value with the minimum positive singular value. So this is the formal definition of our k-value. Here's another toy example illustrating the effectiveness of k-value. In the previous example, we added a perturbation to different theta. Well, in this example, we added a set of different perturbations to both groups and keep the theta fixed. As we can see, the matrix with larger k-value, which is the red one, will tend to have larger quantification error compared to the matrix with the smaller k-value, which is the blue one. We also verify these results using simulated data. Back to the two example genes we started with, we can find that the estimated isoform abundance and the true abundance are more likely to locate near the diagonal of this plot for the gene with smaller k-value, meaning that estimated abundance better represents the true abundance when the k-value is small, compared to this complex gene with large k-value. And for a more general result in the whole transcriptome, the quantification arrows show apparent positive association with k-value across all the genes. And when applied to differential expression analysis, the F1 score decrease as k-value increase, implying increased quantification error. And using real data set for the over 17,000 biological samples from 3 consortia, we find that the quantification error increase with k-values in almost all the samples. This also verifies k-values. Besides computational results, we proved by mathematical inference that the relative quantification error is approximately bounded by the multiplication of k-value and the weighted sum of the relative perturbation on B and even A matrix. So as a conclusion, we designed k-value as a robust index of gene isoform quantification error and validated its performance using simulated data, a huge amount of real data, and the mathematical derivations. More results and proof about k-value can be found in our work, mini-quant. Long-race improved gene isoform quantification by reducing the data deconvolution error. Thank you so much for listening.